Good morning, everybody, and welcome to today's webinar on tools to enable a successful bioeconomy. My name is Mark Whelan. I'll be your host today. I'm the Programme Manager for the Technology Gateway Programme in Enterprise Ireland, based in Shannon. This is the third in our series of webinars on technology insights. The idea being to try and give you new insights onto technologies that can help companies to grow both their products and their services. Today's presentation is given by Tim Yeomans, who's the Gateway Manager in Shannon ABC and IMAR. I'll let Tim introduce himself in a moment. But just to say that the format of the presentation is about 25 to 30 minutes, followed by Q&A. You'll be able to see down the right-hand side on your menu that there is a questions section where you can post questions to us. You can do that at any time, and we'll cover them at the end. Also, just to remind people that the presentation and the Q&A session will be recorded and will be available next week on our YouTube channel. So I'll hand over to Tim and let him take away.
Thanks very much, Mark. So I'm just going to switch off my camera there just for the presentation. So you're all very welcome along to the presentation today, which is around technical tools for successful bioeconomy. So just before I get started into that, just wanted to give you a brief kind of a background and overview onto the Technology Gateway Programme. And obviously this worked very well in practice, but it's slowing down now. So this is the Technology Gateway Network map. So the Technology Gateway Network consists of 16 technology gateways all around Ireland, and all based in the Institute of Technology or the technological universities, and they're there to help Irish companies. And they've all got a reasonably similar structure and a similar process and outlook, I suppose, which is to help Irish companies develop more and to earn more revenue. So I'd encourage anybody, any company who wants to get involved with research to contact Technology Gateways. So Shannon ABC, just a few bits about us, I suppose. So we're actually a collaboration between the Munster Technological University in Kerry and Limerick Institute of Technology, and we're established through core funding from Enterprise Ireland. And I suppose our core area in what we work on is in bioresources. And so there's quite a lengthy statement here, I suppose, but the key word here is bioresources. So helping companies and organizations get more value from bioresources, which obviously feeds in very well to the bioeconomy. So these are our technology themes and how we work with companies, I suppose. So bioprospecting, which is looking for high value compounds, bioprocessing, scaling up those compounds, and then analytical and research services, which provides a support structure to the rest of what we do. And we also have a technology theme around food innovation because we do a lot of work with food companies. And I suppose while that looks reasonably technical, what it really boils down to is these kind of three questions here. So helping companies find out what it is, how do they get it, and what can they do with it. And as we go through the rest of the presentation, I'll be referring to these three questions quite a lot just to try and, I suppose, help people get context for different technical tools that can help them. So the bioeconomy is a word that people are probably very familiar with at this stage. I suppose it was really only coined probably a little bit over 10 years ago to describe the industry that was based around biological systems and outputs and biological technologies as well. And while companies might hear bioeconomy and think, am I really part of that? Any company that does anything with a natural resource is part of the bioeconomy. If they use a natural process to treat what they do, they're part of the bioeconomy. And although this is data from 2016, you can see it's a massive sector all across Europe. And I suppose the important thing about putting a name or a title on this kind of a thing is that it helps to manage it, and it helps for people to engage within this sector. So in terms of bioeconomy, that any company that does anything, kind of words that people would have heard over the last while, I just wanted to kind of give a bit of an outline here to this. And really, while this looks maybe a little bit complicated, at the end of this presentation, I suppose the ideal thing is that while projects associated with the bioeconomy and that different companies and organizations engage in, while they can appear quite complex at the end of the project, they started off with the same kind of building blocks in terms of the technical expertise that was required to deliver them. And so the green lines that you see here is the bioeconomy. So you're taking a natural bioresource, you're getting your biomass from it, you're processing it, and you're getting your end product. And this is a linear process. And while you can have sustainability built into this by production of bioenergy and biofuels, for example, it's still a linear process rather than a circular process. And again, in that kind of a linear process, looking at different chemicals and materials that can be taken from processing and looking at production of bio-based products. And where the circular bioeconomy comes into it is where you start to try and reuse those and bring them back into the system. So people will be very familiar with organic recycling, for argument's sake, food and feed, but might be less familiar with the kind of concept of that rather than trying to get rid of something that you're trying to share, maintain, reuse, or redistribute. So rather than recycling, reusing something at the end of its life. And then within all this context, you're also thinking about a biorefinery approach to extraction. So some approaches will just try and extract one particular compound from a biomass, and that's not really sustainable. And the concept of the biorefinery approach is really to try and get as much out of a resource as you can. And so different questions that companies need to be asking, I suppose, in terms of this entire process is, the raw material that they're using, is it sustainable? Do they need to look at something else? And how do they look at something else? What technologies are required to do that? In terms of processing, do they use a new material and old technologies, or an old material and new technologies? And again, how do they measure the impact of that? In terms of the final product, in terms of shelf life and stability, are there things that they can do to improve the shelf life and the stability of the product so that it will last for longer and won't need to be recycled? The product can last for longer and therefore can be used for consumption or whatever else it might be needed for. And then in terms of chemicals and materials, how to treat the material, what extraction conditions are used. And so are these green extraction technologies, they require solvents? Are there other different types of technologies that can be used for extraction, and do they work as well as the conditions that they're using at the moment? And these are all questions that different technical applications can help to answer. And so the way that I'm going to approach the presentation today is looking at 3 different projects that we have in Shannon ABC. Some of them are more advanced than others, and we'll start off with what these projects have achieved and then work backwards to the different technical tools that were essential for these projects to be successful. And so the first one here is biorefinery glass. So this project was coordinated by James Gaffey, who's co-director of the Circular Bioeconomy Research Group in Shannon ABC, and was funded through EIP Agri. And these were the partners then who were involved in the project. And fundamentally, this project was about grass. And it was about trying to make grass more productive. And you might kind of think, well, look, how do we do that? But I suppose people will be kind of familiar at this stage in terms of discussions around agriculture and sustainability and greenhouse gas emissions from farming, which predominantly come from cattle. Now, there's an argument there about how real that issue is or not, but nevertheless, cattle normally take in more nutrients than they actually require, which is why the nitrogen ends up as greenhouse gases coming out of the cattle. So is there a way to address this? And so in essence, what this project looked at was at juicing the grass, squeezing the grass. So you end up with 2 products from the grass rather than 1. So you've got this grass juice, and you've also got this press cake, which contains enough nutrients to feed the cattle and for them to be productive. And so there's a company based in Holland called Grassa who have built this grass biorefinery facility. And it's a mobile facility that can be brought from farm to farm. And in essence, what happens is the fresh grass is brought to the machine. It goes in one end, it's juiced, and you get your press cake and your pressed juice. The press cake goes on to be a silage for ruminant feed. And then you get a variety of other different compounds which can be used for a variety of different applications. And so this mobile biorefinery is brought around Ireland for a period of time in 2019 to show it to the farmers in Ireland and to see what they thought of it, because it's all very well to be doing the research and think this is a good idea, but at the end of the day, it needs to be taken up by the people who need to use it. And so there were a number of different trials that were run around the Southwest demonstrating use of this grass biorefinery product facility and showing farmers the different things that could be produced from it. But fundamentally, if you're going to be changing how somebody is going to approach something, you need to demonstrate that it's as effective, if not more effective, than what's there already. And so this is so the flow diagram of what's happening with the mobile biorefinery. And if you go back to think about those questions about what is it, how do I get it, and what can I do with it, you have the what is it here, so the protein concentrate, the whey, the oligosaccharides, and how they get it is through protein separation, filtration, and reverse osmosis. And what they can do with it then is these 3 products that are produced from this mobile biorefinery, which is a protein product for feeding the cattle, a soy protein, so that's from the liquid material, a prebiotic fiber, and a fertilizer. So think about those questions. What is it? How do I get it? And what can I do with it? So working backwards from that end product this project has been able to develop in terms of what is it. It's equipment and high-end analytical equipment like atomic absorption, ICP-MS, HPLC, and also biochemistry techniques. So ICP-MS is a very powerful analytical tool that can measure a range of different minerals and heavy metals. So for those of you who don't remember your periodic tables from school, you've got sodium, magnesium, calcium, manganese, iron, copper, zinc, and so on. All these really, really important minerals and heavy metals. And ICP-MS will measure all these different types of heavy metals and minerals to very, very low levels. You're looking at parts per billion and sometimes down to parts per trillion in terms of analytical sensitivity. And so the way that an ICP-MS works is that you have your sample preparation. So you take your press cake, for argument's sake. It will need to be digested in some way, and you can use a microwave digester for that. It goes into a vessel and into the ICP-MS. And the real, I suppose, advantage for the ICP-MS is a very user-friendly piece of kit. A lot of analytical tools require a lot of expertise in terms of running them. And I'll talk about liquid chromatography, mass spectroscopy later on. But a lot of them need highly trained individuals. Now, not anybody can run an ICP-MS, but it is very user-friendly to run. And at the end of an ICP-MS run, then you will get a readout with a large portion of all these heavy metals and minerals, a readout of what level they're at in your product. And then you can start to work out, do comparative analysis on what grass is like before the press and then after the press, and determine what the levels of these nutrients are in there, which is very important. Obviously, you're going to be feeding that to cattle. So this is a liquid chromatography mass spectroscopy. So it's in essence, it's a HPLC linked to a mass spectrometer. And so in terms of HPLC, high pressure liquid chromatography, the way chromatography works is it separates out compounds according to size. So you've got 3 different types of compounds here. They've all got a different size and they run through this column at a different rate, and therefore they come out to be detected at a different rate. And the way that works in a kind of a process flow is you have a solvent which pushes the sample through the HPLC column. So your sample goes in here at the injector, the solvent pushes it through, and it goes through this HPLC column, which for all the world is very similar to this, and compounds separate out at a different rate. And as they come out, they're measured on a detector, and what you get is this chromatogram being produced as you're trying to detect your different compounds. And so for the grass biorefinery project, they were very interested in prebiotics, and I'll talk about prebiotics a little bit in a while. But in essence, they're sugars. And so you've got a number of different sugars here that were able to be detected by the HPLC because they come out at different time points. And the time point that it comes out at is more or less a fingerprint of the chemical. So you see glucose coming out here, and then 2 ketones coming out here. And depending on the intensity at which they're measured, you can determine what the concentration is of each of those sugars in your product. And again, in terms of comparative analysis for this project, it's very important, but HPLC is widely used for a range of different types of compound to measure what's in there. And for companies to change over from one process to another, or one extraction technology to another, you need to prove that what you're getting out at the end is as good as, if not better than the original technology. So in terms of the how do I get it question, so you can have physical separation, you can have filtration and precipitation. And I suppose part of what I want to talk about today is that for some companies, while some of these technologies might appear quite complicated, some of them are quite simple as well. So filtration is basically you've got holes in a product, and some things will go through that are small, and some things will remain on top of the filter. And so a common piece of kit that we'd use in the lab is this ultrafiltration piece of kit. So you have your filter here, your feed goes in the top, and anything larger than the pore size of the filter will stay on top, and everything smaller will go through. And in this way, you can start to determine what size of compounds are there that can be important for the activity of your compound. So different proteins will have different efficacy and different relevance at different sizes. And also you may not be interested in the small proteins. It may just be the larger proteins that you're interested in. And so you want what's up here, and then you can get rid of what's down the bottom or try and valorize it in a slightly different way. And this type of system is fully scalable. So as companies go to scale up filtration, that there's units that they can get in terms of ultrafiltration and nanofiltration that can be scaled on site at a company. And so the other thing that I just wanted to talk about briefly is precipitation. So again, people may consider that some scientific approaches are quite complicated. And so just in terms of precipitation, this is a very brief YouTube video I wanted to show. This is a dairy waste, which is a protein in there. And these scientists are just adding an acid in there to precipitate out the protein. And what happens here is the acid either slightly unfolds the protein, or it dehydrates the water. But the effect of both those things is that the protein starts to fall out of solution. When the protein starts to fall out of solution, you're then able to take it and use it for other things. So you see the protein coming out here, and it falls down all the way down into the bottom of the flask. And if you think about scale up here for the food sector or something like that, you can use a food grade acid to precipitate out proteins. So you could use something like citric acid for argument's sake. And as you precipitate that out, you've got all this liquid at the top here, which can be decanted. And you've then got a very functional product in a very easy and quick way. And so while some of the technologies that I'll talk about are quite complex, some approaches can be very straightforward as well. So in terms of what can I do with it? And so as I mentioned earlier on, it's all very well to talk about different processes and different ways of doing things, but at the end of the day, it still has to be effective. And so for the grass biorefinery project, for the press cake, was that going to be as effective for feeding the cattle as silage was? And so an animal trial was required for that, which can be quite a kind of, I suppose, an undertaking, and it was done with UCD. But there's lots of really good capability all around Ireland in terms of animal trials and feeding trials, particularly in the dairy sector. And so for companies who are interested in this, there's lots of resources there for them. And so sample size in terms of, how many cattle you're going to include in the trial and how you're going to measure your endpoints is very, very important. And it's that identification of endpoints which really dictates which kind of technology you're going to use. And so this table here summarizes the impact of the press cake. So you've got grass silage here, which is what's being used at the moment. And then you've got the press cake here. And so if you look at these key endpoints, milk yield, fat, protein, milk solids, they're all very similar between the two. And different biochemical methods would have been used to measure these things like fat and protein. And what's really important here is that you're in essence making two products from the grass, but for one of the products, you're getting as much productivity as you are from using the entire grass process itself. In terms of feeding the feed from the liquid byproduct to pigs, that showed an increase in yield. And the project were also interested in prebiotic activity, which I'd like to talk a little bit about now as well. So people may be familiar with probiotics. So probiotics are the good bacteria that companies like Yakult will include in their products, and Actimel as well, although you won't see probiotic on any European or product sold in Europe now, because probiotic is considered a health claim and is not approved for use in Europe. Prebiotics are the sugars that probiotic bacteria like to grow on. And the benefit of using prebiotics is that you can encourage that good bacteria growth. So this sugar here is glucose, it's a monomer, and all bacteria like to grow in glucose because it's very, very easy for them to eat. So E. coli and Salmonella, which in inverted commas bad bacteria, will grow very well on it. Bifidobacteria and Lactobacillus, which are probiotic bacteria, will also grow very well on it. When you've got a prebiotic sugar like Nistose, it's a much more complex sugar. And E. coli and Salmonella don't have the capability to grow on it nearly as well as Bifidobacter and Lactobacillus. And so where you can prove that a sugar is a prebiotic, you will select for these kinds of bacteria to grow in the gut of the animal and therefore to make it more healthy. And so the way that we go about this is that we would grow the prebiotic bacteria in a liquid culture. And so we'd have a growth in there with just the prebiotic as a sugar source. And what you'd expect to see then for something like Bifidobacter is this growth curve here on a prebiotic sugar source. So it grows really, really quickly here. What you'd expect to see for E. coli is that the growth curve tails off. It grows a little bit, it can chunk down some of it, but not really too much. And so you're going to selectively grow the probiotic bacteria much more effectively in the animals. And in the studies that the project did on how that worked, they saw that compared to commercial FOS and inulin, which are both commercial sources of prebiotics, the grass FOS performed as well as, if not better, than some of those prebiotics. And so microbiology from a sort of technical perspective can be really useful to help companies make decisions about how they want to progress with a certain route for involvement in the bioeconomy. And then the final area that they would look on for this project was a biofertilizer. So NPK, really, really important for nutrient content. So this is the NPK value of slurry and the NPK value of the biofertilizer that was produced by this project. And again, you see that it's very similar in terms of the NPK values, and ICP-MS would have been used to determine these values. So when you look back at this big, really complex piece of kit, it all stemmed from answering those questions around what is it, how do I get it, and what can I do with it? And I really encourage companies that while some involvement in the bioeconomy or trying to be more sustainable or whatever it might seem like a really big thing to address, it all comes down to asking those three questions, identifying the technical tools that can help you to answer the questions, and then to go on to really develop something that's impactful. So the second project I want to talk about it's only ongoing about 4 or 5 months. So it's being led by Athlone Institute of Technology, and Shannon ABC and LIT are partners in the project, led by Catherine Collins and Paddy Murray. And this project is about degradation of plastics, and in degrading the plastics, to use the breakdown products as we degrade them to produce more products and bioproducts. So people will be very familiar with the problems associated with plastics contamination globally. Plastic particles are basically ubiquitous around the environment at this stage. And the big problem is that a lot of the products that we eat and that we use have these kind of really difficult to degrade plastic materials. And so this project, the Bioiso project, is really made up of 2 projects. So The one that we're involved in, Bioisop, is this bit here. And as we produce these monomers, hopefully they will then progress onto a different European funded project, which will take these monomers and use them as building blocks to build other bioproducts. But this is what we're really interested in here, is this breaking down of the plastics in the first instance. And so we go back to the three questions again about what is it, how do I get it, and what can I do with it. So in terms of breaking down plastics, we're using fungi and enzymes. And the way that we start off with that is from a microbiological perspective. So these are just two plates with fungi growing on them. And there's a particular biomass built into the media of this plate. And we want to see if the fungus can break it down. And if you see this brown shadow around the fungus growing down there, and the yellow shadow that's growing around there, that means it's producing an enzyme that's breaking down the biomass. And as we know it's doing that, we then know that that fungus can produce an enzyme to destroy that biomass. And so we'll take that fungus, we'll use our microbiology capabilities again and grow it up in a liquid media culture, because we want to isolate that enzyme. Because as we isolate the enzyme, we can then start to make an industrial process from it. So in order to do that, we use a procedure called electrophoresis. So in essence, this is a separation technology again. So you take your sample from your media and you apply it to this gel, and it separates according to size. And so as the sample runs down through the gel, you'll get different bands for the different proteins and enzymes, which are separated according to size. And so you can use one just for your media, and then one for your media and your fungus, which produces the enzyme. And you'll hopefully see a different band in this lane than the other one, because then you know these bands are associated with something that's being produced in that media that's not being produced when the fungus isn't there. And so then you can take those bands and you can go to our ultrafiltration that we spoke about before, because you know what size those bands are. They're 116,000 Daltons. And based on knowing what size they are, you can use a filter here and start to concentrate those proteins. And once you concentrate those proteins, you can then go back to our HPLC and to our LCMS, try and identify what those proteins are. And all enzymes are proteins. And so as you know what they are, you can then start to develop an industrial process for production of those enzymes. And so in terms of how do you get those enzymes, how do you get to that stage? So we have a fungal biobank and a microalgal biobank in Shannon ABC. And while most people will be familiar with fungi, you won't be as familiar with microalgae, I'd imagine. So microalgae are pretty much the scum that you see growing on ponds, so green scum in essence. But if you grow them in the right conditions, they can produce really interesting compounds. And so we're going to take those fungi and those microalgae and try and apply them to degradation of plastics. And the biggest problem around plastics is this structure here. So the way the polystyrene is produced is you take a styrene monomer, you polymerize it, and it forms this structure here, which is really difficult for fungi and for all different types of enzymes to break down. What this project will do is it's bring a novel green sonic technology to try and break up these polystyrene compounds into smaller groups to make them more to be more easily attacked by the fungi and the microalgae. So as they're broken up into smaller bits, they become more easily digestible and degradable. And so once we have that, we can use our biobanks of fungi and microalgae and apply them to the degradation of the polystyrene smaller particles, and hopefully break them down into monomers. And as they're broken down into monomers, we can then start to send them on to the second part of the project, or have a success rate for the Bioisop project, and move on then to the production of bioproducts with other partners. And so in the detection of these monomers, we'll be going back to our analytical equipment again. So our LCMS, our HPLC, and our GCMS as well. And so while HPLC is good for looking at say solid or liquid compounds, GCMS is gas chromatography, and so you're looking at vaporization. And in terms of plastics, it's a really useful way to determine whether styrene is in its monomeric form or its polymeric form. And so these will be used to be able to kind of generate success or not of the project. So the final project I'm going to talk about today is one with Galco Steel. So Galco Steel are a company based in Dublin and sites around the country as well. And they do hot dip galvanizing. And so they may not be kind of an immediate candidate that people might think about for the bioeconomy, but they're taking a lead in this area through some research that they're engaging in. So through the hot dip galvanizing process, you get a lot of surface water that's contaminated with heavy metals, so things like zinc and iron and so on. And that needs to be purified before it can be released to the environment. And the way the Galco used to do this was using ion exchange filters. Now, ion exchange filters are expensive, and in terms of recycling, they can't. They're just dumped. And so Galco Steel wanted to determine whether there were some options there about approaching this in a more sustainable way. So while they were doing this, a company in Scotland had developed a biofilter based on spent grain. So spent grain is produced from the fermentation of grains in distilleries and in breweries, and it's reasonably well established that spent grain will absorb heavy metals. And so this Scottish company have produced drams that are kind of a similar size to the ion exchange systems, and activated the spent grain to make it make it absorb heavy metals in a very effective kind of a way. And Galco Steel have these drams on site at the moment, and they've had audits from the EPA over the last while, and the EPA have been very happy with the data that they're seeing in terms of the water going into the municipal waste treatment sites. And so Galco came to us because that's great. They have this linear process now in terms of going to spent grain, and that's great. It's part of the bioeconomy, but it's not really circular, because if they haven't got a use for the spent grain, they're still going to be dumping it. And so they came to us to have a chat about, again, what is it? How do I get it, and what can I do with it? And so primarily it's looking at the heavy metal content in the spent grain, because that's really what it's absorbing. So going back to using our ICP-MS and looking at the heavy metal content in the spent grain, and then based on what's in the spent grain, trying to identify applications for it, which might be as a soil amendment product, it might be an animal feed, and going back to our animal feed trials again, or it may be as a feedstock for biotechnology applications, so going back to microbial capabilities. So we've completed a feasibility study with Galco Steel, and we're planning to submit an innovation partnership later on the year. But what we're really kind of excited about in terms of applying to this project is high performance thin layer chromatography, which is a new piece of kit that we've got recently enough through the EI equipment call. And so the way that thin layer chromatography works at its most simple is, it's chromatography, which I kind of mentioned earlier on. It's about separating out particle sizes. And so you have a glass plate, which is covered to all the world with what's like, it looks like icing sugar, I suppose, solid icing sugar. You apply different compounds to it. You have a solvent here, and the solvent will drag the compounds up through the plate. And as it drags them up through the plate, they'll separate out according to size. And as they separate out according to size, you can use a standard to try and determine what those compounds are. Now, this is a very basic version of it. For the system that we have in Shannon ABC now, the HPTLC, it's a very advanced version. And so as you run a sample through it, you can use a densitometer that's based in the software to determine what the concentration of this band is versus this band. So if this is your standard that you run, you know what the concentration of this band is, and therefore you can calculate the concentration all across the board. And you can use this for a range of different applications, and we'll be looking at it in terms of, say, protein and carbohydrate that are in the spent grain, because it's really important for how well they absorb the heavy metals. But the HPTLC can be used for a wide range of applications, and looking at cannabinoids in hemp. You can look at active pharmaceutical ingredients, and the advantage around it is that compared to LCMS, which needs quite a lot of solvent, it can be a little bit more time consuming. We can run a lot of samples all at the same time using HPTLC and use less solvent. So it's a very rapid method to get results very quickly. And so you can screen through a lot of samples reasonably quickly for a company. And then when you need to get really, really down to a much more sensitive level, you can then use a HPLC or an LCMS. So I suppose the fundamental questions that I wanted to get to in terms of the technical capabilities around what is it, how do I get it, and what can I do with it, it's really to encourage companies that while there may be really big picture questions that they need to address, and it sometimes can seem quite difficult to do that, when you work it back with just those three questions and what the answers are that you need, there are technical resources there that are available to you to assist you with going down that road in terms of sustainability and circular bioeconomy. And so that's all I have for now, and certainly happy to take any questions.
That's great, Tim. Thanks a million. So a couple of questions starting to come in. So the first is, how long does it take to do those types of chemical analysis?
It depends to a certain extent on the number of samples, but for HPTLC, you could have a result within a week if there's a method set up on it. If we need to set up a method, it can take a little bit longer. For ICP-MS, it's very quick. You'd have that done in a day. And so it really depends on what it is that you're looking for, I suppose, to a certain extent.
Okay, and a follow on from that, and that might have answered it. Is it feasible for a farmer, I suppose, companies to do those types of analysis themselves, or is it always the case that it needs to be sent to a lab like Shannon ABC?
I suppose the pieces of kit there they've been talking through can range anywhere from 100,000 euros up to kind of 400,000 or 500,000 euros. So they're expensive pieces of kit. And so in general terms, it would be, if it was an SME or something like that, they certainly may not have the resources to purchase that themselves. And if it's not a thing that they need consistently, it really wouldn't make sense to purchase it anyway. And I say lots of different third level colleges all around the country have this kind of kit. And so I absolutely encourage companies who want to engage in this to contact the Technology Gateway Network, but also their local university or IoT or TU to see if they can be helped in this way.
Okay, so maybe a follow on from that then is, how expensive is it to run those actual tests, and how can you set up a project for a company to ensure, for want of a better word, they're getting the best kind of value from that they're getting answers as quick as possible, so they're not waiting for lots and lots of tests to find out, well, maybe this didn't work, or maybe it's not as
Yeah,
valuable as I thought it was.
yeah, absolutely. So I suppose in the first instance, we kind of sit down with the company and we have a chat through what they want to do, and we've got a lot of experience of doing this. And if we'd look at it, I suppose, from a knowledge-based perspective first, so we look at the raw material or the byproduct it is they want to look at. And if they're talking about trying to extract something that really won't be commercial at the end of it, or trying to identify it, we'll have that conversation with them to try and de-risk any subsequent work based on, look, is this really something that's worth proceeding with or not? And look, sometimes you're not going to know until you do it, and that's just part of research as well. But we do try and approach to de-risk it as much as possible. And once we've done that, we work out what kind of a way, I suppose, the budget for it in the first instance. So, can we apply for an innovation voucher, as a contract research, whatever it is, and trying to develop the project around the budget that's available for it, and to try and get the best answers we can within the budget that is available, I suppose. It's not necessarily cheap to do some of this analysis, but we would try and then work with companies maybe to develop it more in a kind of a modular fashion. So get answers to question A first, and once we have that question, then it makes sense to go on to question B.
Great. Sorry, could you turn on your camera, please? Just we
Oh yeah,
can't
sorry,
see.
yeah.
Yeah, no bother. So, I mean, you've talked about state supports available. So maybe could you just give people a quick overview of how a voucher works, or how it might work, or an innovation partnership, or even SFI?
Yeah, sure. So innovation vouchers are very straightforward, really. So a company will go online, they'll create a username and password and fill in the form that's available there. And I suppose the innovation vouchers, every limited company is more or less eligible for two €5,000 innovation vouchers and one co-funded voucher where EI put in half and the company put in half, up to a total value of €10,000. So it normally takes somewhere between 2 and 4 weeks to get those approved. And then you can set up a project with a research provider to do the project. The IP belongs to the company. VAT has to be paid on the innovation voucher as well. And I suppose it makes sense where possible to engage with a research provider before you put in the application, just so that you've got in terms of expectation management around what can be delivered for a voucher. But they're a really good way for companies to kind of get a foot in the ladder in terms of engaging with a research provider. Innovation partnerships, I suppose, are the next step up from that, and I think are kind of, I'm not sure what the lower limit is, around 20-30k, up to kind of €250,000. Enterprise Ireland will fund up to 80% of the direct cost of those projects, depending on the size of the company and the type of research. And you need to be a client of a state agency to get access to the Innovation Partnership Fund. Okay. And again, if you're not sure about that, suggest you contact one of your technology gateways or Enterprise Ireland and the Innovation Partnership team.
Okay, thanks. So another one coming in. So are there certain types of waste streams that this is more suitable for? So I suppose we talked about dairy, but obviously the steel wouldn't have been one you'd have thought of. Is it more amenable to some industries than others, I suppose?
Yeah, I suppose the technology we work with, I suppose, are predominantly platform technologies. So they could be applied to any sector, really. Some byproducts or waste products are easier to reuse or recycle, I suppose. So if they're coming out of the food sector, if they're, depending on how they're categorized, so if something is categorized as a class 3 waste, it's very difficult to put it back into the food sector or cosmetics, for example, and you might be looking for a different kind of an application for it. So it does depend on the categorization of the waste or the byproduct, and if you can avoid it being categorized as a waste, even better. But I suppose the technologies that can be used for this are not sector specific. They'd be platform technologies, I suppose.
Okay, great. So are there any other technologies that can be useful for biomass characterization?
Yeah, sure. So calorimetry is one. So looking at the calorific value of different products. So particularly if you're looking at things to be to be burned, for argument's sake, you need to know the calorific value of it and how effective it's going to be. We can look at, say, how different byproducts can be used using cell culture. So we can grow mammalian cells in the lab. We can add in a product to it and see what it does to the cells. Does it kill them? Does it make them produce a certain compound? Does it have an antioxidant activity? So we can measure that kind of an approach. And then something like FTIR, so that's an analytical tool. So we do have that capability in Shannon ABC, and a photonics approach can be quite helpful. And one of our other technology gateways, Kappa, have a lot of photonics expertise. That's their area of expertise, which can be useful in terms of looking at biomass and what it can be used for.
Okay, and a follow-up to just one of the early ones. So what are the other ways that you can actually identify the applications for the biomass or the waste product or the byproduct?
Yeah, so again, I suppose it's really finding out what's in there in the first instance. And if it's the biomass that we don't really know a huge amount about, we'll start off maybe looking at, the top level things that are in there: proteins, carbohydrates, lipids. And once we get an idea of that, it might give us a better kind of steer on which sector it might need to go into. And again, talk about a knowledge-based approach. Again, you're going to the literature and seeing what's already being reported to be in those kinds of products to inform what you might try and do with it. And again, going back to the cell culture capability that we have in Shannon ABC as well, that would be another way to try and identify where a particular byproduct or waste product might be appropriate for.
Okay, so the company don't need to come to you with, for want of a better word, the intended solution or the intended use.
Yeah, no,
You
absolutely
can
not. I
help
mean,
them discover what that might be.
yeah, we get a range. Some companies will come with a very specific idea in mind in terms of, look, we think this is an idea here. We want to have an application in the cosmetic sector. Can you test this for us? And that's absolutely fine. And other companies will come and just say, look, we have this waste product. We don't know what to do with it. Can you help us? So it really goes the full spectrum in terms of the knowledge that the companies may have or the ideas that they may have, I suppose.
Okay, so I just remind people that you can use that questions tab there to send more questions to us. So another one, are prebiotics actually effective?
Yeah, look, I mean, they certainly will selectively help those probiotic bacteria to grow. Prebiotics are not approved for any health claim in Europe in terms of human applications, but they certainly can have benefits in terms of supporting the growth of the beneficial bacteria. Now, it's important for that kind of an approach. You need that mix of bacteria, and animals do as well. So you don't want to be killing off any particular species, even E. coli. You need to have that in your gut for certain things it produces. But definitely they can play a beneficial role in terms of supporting a healthy gut system, I suppose.
Okay, and are there any particular waste streams then that you can't work with? You say it's more difficult if it's level 3, I think you said. Or...
Yeah. So we have been approached before from some companies, I think in the pharmaceutical sector, with data solvent byproduct. I'm not sure if it's hexane or methanol or something like that. And there's just not a huge amount that you can do with that. So we're happy to have a chat to companies about whatever it is that they need to address. Some can be definitely more difficult than others, and some, like if you've got a certain byproduct that just really aren't good for anything else. I know some companies have looked at developing things like incineration blocks from them, so burning them in essence, because they can't be used for anything else. And they need to make sure that off-gassing and all the rest, that is fine. But in essence, incineration. So there's a company based in Limerick who incinerate manure, and so they have this system that burns manure at really, really high temperature. So it gets rid of everything that's bad. So you're not going to get any noxious off-gassing because the temperature is too high. But from that burning, you can actually heat the sheds that the manure came from in the first place. So there are ways to approach using things that otherwise may not be able to be used. And I suppose all I'd suggest is for companies who aren't sure, just to get in touch and we can have a chat to them about it.
Okay, great. I think that's brought us to the end of the questions that have come through there, unless anybody has any last minute ones just while I'm wrapping up. I just remind people that the webinar will be available online on our YouTube channel next week, and we'll send out that link to everybody who's registered, even if they couldn't make it today. So I'd like to thank Tim for his time and an excellent presentation today, and remind people that we will have our next and our fourth webinar in the series on the 13th of May. That'll be on process monitoring and automation from SMEs and will be presented by the IMAR Gateway. So if there aren't any other questions, I'd like, again, as I say, to thank everybody for the time today. Thank Tim. Sorry, it's a bit of a tongue twister, Tim. And we'll see you all at our next one, I hope. Thank you very much.